When Mira invited me, I appreciated the invitation, and I thought I would talk to you about the work I've done on extremophiles. And as some of you know in our conversations during the break and during our lunches and dinners, and I've been working on other things as well, and I'm happy to talk to you about what I'm doing on the food-water-energy nexus. But today I'm going to talk to you about my lessons from nature. And in particular, I want to talk to you about the lessons I've had from Nature magazine, and talk to you about the concept of molecular chaperones. So in a show of hands, how many people know what a molecular chaperone is? Oh, good, about half of you know about this concept. Well, my story on chaperones starts with Alvin, which is a research submersible that I dove on when I was a graduate student. And I was working on organisms that lived at high pressures, which are the conditions in the deep sea. So if you think about our Earth as a planet, 70% of it is covered by water, and most of the water, the average depth of the ocean is about 3.8 kilometers. And that means there's a very high pressure, low temperature environment in the deep sea. Globally, the Earth is covered by mud, dark, cold mud. Now, there are these rare places where there are geothermal hot springs in the deep sea, the so-called black smokers. And these black smokers, which are at high pressure and high temperature, are unique in the world in that they're not smoke coming out of here, but it's a metal sulfide precipitate that looks black, and it looks just like a smoker. Now, these places in the deep ocean, in total darkness, surrounded by a 4 degree Celsius ocean, are sometimes surrounded by diversity of interesting organisms. And these organisms are living around the black smokers the same way we would be surrounding a fire if we were sitting at a campfire. Now, when I was a graduate student, there was an outrageous microbiologist named John Barrows, who had samples from one of these black smokers and speculated that life could grow under these high pressure conditions, also at extremely high temperatures. Now, he published this in Nature when I was a graduate student, and I knew a lot at the time about the effects of pressure on biological systems. And this paper, which appeared in Nature in 1983, was an incredible wow. Because 250 degrees, if there's any chemists in the auditorium, they wouldn't understand that this is a biological outrage. I mean, a chemical outrage for biochemistry. So I went on to show that this paper was wrong. So not everything that you read in Nature is correct. In fact, I published a paper in Nature called Possible Artifactual Basis for Apparent Bacterial Growth at 250 degrees. That was a polite way to refer to the black smoker bacteria as BS bacteria. So it was wrong. But it led me in my career to start studying extremophiles, organisms living in high temperatures or high salts or low temperatures and high pressures. And I put this slide in instead of the movie because the movie wouldn't run, but also because tomorrow we're going to talk about aging, and you have this great example of me. So there are some very extreme environments and we're familiar with relative to what we consider normal. And obviously the organisms that are living in the Dead Sea and in other extreme environments, they consider that a normal condition. So the halophiles, which are, for example, inhabiting the salt flats around the Dead Sea, or as we heard this morning, in places where the salt concentrations are okay, in the bottom of the deep sea, these organisms are interesting and important to think about in terms of what life is capable of adapting to. Life as we know it now is divided into these three categories: the bacteria, the eukarya, and the archaea. And the most extreme in organisms that we know are in the archaea. Now, when you think about life forms, the archaea and the bacteria have some very unique and interesting features. And one of those features is that they are very small. So this is a picture of the hair on the back of your hand. And if we zoom in on that hair, at this magnification, that's a yeast cell, and these are the bacterial cells. So these very small organisms, which are the most successful organisms on the planet in terms of their overall survival from the 3.5 billion years that they've been here, these organisms are sort of the ultimate biological machine. And this machine runs on the nanoscale. They're made up of 70% water, 15% protein, 6% RNA, 3% miscellaneous, 3% carbohydrate, 2% lipid, and about 1% DNA. So in terms of the biological composition, they're mostly protein. This is not so different than us. We're about the same distribution of liquid, i.e. we're mostly water. If you put a human into a blender, we have about the consistency of yogurt. But inside the human organism, things are organized in a different way than the microbes. Looking inside a microorganism like a bacterium or an archaeon, you see the membrane in a 100 nanometer cube is made up of these blue blobs, which are proteins, this red stuff, which is lipid, and polysaccharides. And inside the cell, we see these yellow ribosomes and some blue structures. They're proteins. And in a 100 nanometer cube, there would be about 450 proteins. These green things are tRNAs. There's about 320 of them. And there's some ring proteins that occur also within this volume. This region where the nucleus is, or the nucleoid in a bacterial cell, is a DNA wrapped around, which looks like ropes here, wrapped around proteins. But this picture is misleading. And that is because this, just as you see in this picture, a still image of something that we know is very mobile. Inside that microbial cell, transcription and translation are going on. The DNA is being made into RNA at a rate of 70 nucleotides per second. And the RNA is being made into protein at the rate of translation of about 40 amino acids per second. And everything is bouncing around. So a single protein, 40 kilodalton, will diffuse or move from one end of the cell to the other end within about a second. It'll go there and back and halfway back again. So everything is in constant motion. This system is incredibly dynamic. Within the central dogma of the molecular biology, we know that DNA replicates. Transcription is the process where DNA makes RNA. Translation is the process where RNA is turned into proteins. And we know sort of how this process works. We also know that Ada Yonatou, who was mentioned yesterday, got her Nobel Prize for solving the structure of the ribosome, which is this machine within the cell that turns the RNA information into a polypeptide chain. And that polypeptide chain spontaneously folds into a functional protein. And Christian Anfinsen got a Nobel Prize a long time ago for showing that all the information for protein folding is contained within the amino acid sequence. That is to say that the polypeptide chain spontaneously folds into its functional form. But there's a problem with this idea. Anfinsen did his experiments in an infinitely dilute solution. And yet I just told you that the inside of the cell is not only cluttered with material, crowded with proteins and RNA and ribosomes and DNA, but also that it's incredibly dynamic. So how do proteins fold inside the cell? Not the question of what Anfinsen solved the problem, that the information is available in the polypeptide chain in an infinitely dilute solution, but how do proteins fold inside the cell? Well, the answer to this question came, at least in part, in the early 1980s with a theory called the molecular chaperone theory, which is what I posed to the audience, and half of you know what this is. The molecular chaperone theory says that inside the cell, protein folding is mediated by a special group of proteins called molecular chaperones. And the newly synthesized polypeptide is escorted to these ring structures which exist inside the cell. And inside these ring structures, protein folding occurs. And John Ellis in 1987 published again in Nature, in the crowded intercellular environment, molecular chaperones prevent inappropriate interactions between nascent polypeptides and allowing folding to occur. This was a really important theory, and since has evolved and become very well accepted. The concept that protein rings are called chaperones has gained a lot of momentum. Electron micrographs of the proteins from bacteria show a 7-protein ring that looks like this in cross-section, and people then speculated how they might function for other proteins to fold inside these chambers. The problem with this theory was that we had lots of evidence for bacterial versions of these things, but we didn't know what was going on in the cytoplasm of the eukaryotic cell, and we had no idea what was going on in the extremophiles. So let's go back to my going into the extreme environment. One of the first questions I asked as a postdoc was, do the extreme organisms also have these ring structures that might be involved in protein folding, or do they do something quite different? I was studying an organism from Japan in Beppu, and it grew at 85 degrees and pH 2. The organism is called Sophilobus shabbatai, and it's a small organism. It's about the same size as a bacterial cell, and sure enough, it has a ring structure. But the ring structure has 9 subunits in the ring. And when I sequenced the gene that codes for the protein, when I isolated it, it looked like the E. coli protein, which is called GRO-EL, and they're all called chaperones. But it has these 9 subunits in the ring. It looks more or less the same. But the sequence was different than the bacterial proteins. And all of the bacterial proteins were highly conserved. But the Sophilobus protein, which I called a Rosetta-some, to play on the idea that it might be a Rosetta Stone, the Sophilobus protein was not related to the bacterial ring proteins, but it was 40% identical to a mouse protein. It was related to a protein called TCP1. TCP stands for tailless complex polypeptide 1. So this was a big deal, and I published this. I had a cover on the issue of Nature, and it was a chaperone from a thermophilic archaebacterium is related to the eukaryotic protein T-complex polypeptide 1. So I was famous for about 15 minutes or so, but what happened was I looked closely at what's going on. And this protein showed me that the concept of molecular chaperones is wrong. That this idea is fundamentally flawed. The Rosetta-some was related to a protein called TCP1, and TCP1 is a membrane protein in yeast, and in all other organisms it's related to membranes. In Sophilobus, it's also a membrane protein, as shown by these immunofluorescence images. And when you look at it in electron micrographs by gold labeling sections of cells, you can see these black dots indicate where the protein is in the cell, and it's a membrane protein. Well, for me, this was a light bulb going off, because Sophilobus lives at a very low pH. The inside of Sophilobus is 6.5. The outside is pH 2. So these organisms have to cope with an enormous concentration of protons on the outside trying to get in. And this protein that we're looking at is a structure that helps to stabilize the cell membrane. So the concept that that these chaperone proteins are involved as a ring structure folding other proteins was just a little bit misguided. That in fact the interior cavity of these cells, when you consider more detail, can only accommodate really small proteins, less than 40 kilodaltons, actually less than 20 kilodaltons, which means that most proteins, the average size is 40 kilodaltons or higher, can't fold within this cavity. And the notion that chaperones are somehow useful is actually misguided. The great tragedy of science, as Thomas Huxley said, is the slaying of a beautiful hypothesis by an ugly fact. So when you try to replace it in theory that's been well accepted, you have to propose something else, and I propose the sensitive chaos theory for how proteins will fold in vivo. And in this theory, the inside of the cell protein folding is not mediated by specialized proteins like chaperones, but by random molecular interactions enhanced by crowding and the conditions inside the cell. And the conditions inside the cell are formidably complicated. But the model suggests that in the cell, in the sensitive chaos theory for protein folding, that in the cell molecular interactions are inevitable. They're random, they can be positive or negative, they can be productive, or else the system just recycles its mistakes. The image of a dance floor comes to mind, but not this kind of organized dance floor, but rather this kind of dance floor where this man is dancing with a woman standing over here, and it isn't obvious from the picture. So how do proteins fold inside the crowded dynamic cell? I think we learn a lot from the extremophiles. The molecular chaperone theory is, as half of the people in here raise their hand, well known and well accepted. It's in all the textbooks. The sensitive chaos theory, which I just told you, is now known by the number of people in this auditorium more than it was known before. I published it many years ago. It's roundly ignored. But the concept that this is what's going on inside the cell is an interesting one. And we learn a lot from nature. We also learn a lot from extreme environments. The question is whether scientists don't solve problems and only colonize them, or whether or not we're going to, in this institute and other institutes of scientific higher learning, going to keep the open mind required to move on when the ugly facts arise. So I'll end here. Thank you. Yes, I haven't heard it, but let's talk about this after line. The interesting thing about the sulfolobus membrane, in fact, all of the archaeal membranes, is that they're very different than the bacterial membrane. So the bacterial membrane is a bilayer, and it's like a soap bubble. It's moving around the cell. The sulfolobus membrane, in order for it to be impermeable to protons, is actually a monolayer. It has not a lipid chain that interacts as a bilayer, but it has a solid chain that's a C40, it's carbon 40 chain length, and it just vibrates. And this protein, which we've shown now very categorically, binds to the probably very inside of this membrane and limits its mobility, and thereby limits the flux of protons into the cell, which acidifies the cell. The problem with this theory is that nobody believes me. And even though I published in the Proceedings of the National Academy like 15 years ago, I have yet to be invited to Stockholm. So I don't understand this.